everyone. Today we're going to be discussing Chapter 7, which is Lymphocyte Signaling. So why is signaling important? So signaling essentially links a signal from outside the cell to a reaction inside the cell. So this is bridging across the plasma membrane. And this is particularly important for ligand binding to those receptors. So today we're going to be discussing mostly T and B cells today. And instead of focusing on all the names of the different proteins, I'd like you guys to try and focus on some of the themes that we talk about. So like I said, receptors convert extracellular signals into intracellular events. And in particular, this uses protein kinases, which are enzymes that covalently attach a phosphate group to a protein. And this is called phosphorylation. Typically, this activates proteins, although it's possible in some instances that it represses protein activity. And we'll be talking today mostly about tyrosine receptor kinases, which are specific to phosphorylating tyrosines on proteins. And this helps create new binding sites for different proteins, which will help propagate the signal. And phosphatases, conversely, remove phosphate groups. And this typically switches a protein off for similar reasons to how the kinases turn proteins on. So we'll be looking at those receptor kinases. So the kinase domain is typically inside the cell. So here on this gray line you have the plasma membrane. So there's an extracellular domain here, and this is linked to a tyrosine activity. So a receptor tyrosine kinase is when you have both of these domains in the same protein. So here, when the ligand outside the cell binds to the receptor, this can cause dimerization. This dimerization then allows this kinase activity to occur. So the activated kinases will then phosphorylate downstream signals. It's also possible that there are receptors that are not tyrosine kinases. So in this case, the receptor domain is linked to an intracellular separate kinase domain. And here you can see this represented. So in green, we have a separate kinase protein that's different from the blue extracellular receptor. Again, though, when a ligand binds the receptor, these receptors dimerize, and this similarly allows kinase activity to occur. So one thing we'll be talking about today is the idea of signal strength. So receptor activation and downstream signaling may occur over a minimum threshold. So this is a certain level of a ligand where above that level signaling can occur. And signal strength refers to the amount of signaling due to the amount of ligand. And a few different factors can influence this, including ligand affinity, ligand abundance, and concentration of intracellular signaling components, which we'll discuss. And some responses might be binary, just a simple on or off in the signal, whereas others might change strength with the signaling strength. So here you might get different levels of signaling. So intracellular signals are also mediated by multi-protein complexes. And this is through protein interaction domains or modules. And this links back to that idea of phosphorylation by tyrosine kinases. And this creates binding sites for proteins to come together to form multi-protein complexes. And one really important domain is this SH2 domain, or circumhomology 2 domain. And this is a particular amino acid sequence, it's only about 100 amino acids long, where a phosphate can be added. So the kinase will recognize this specific amino acid signal and will add a phosphate to this domain. So one example of these multi-protein complexes are scaffold proteins. So scaffold proteins are large proteins within a cell that, when phosphorylated, bind to multiple different proteins. So they bring this signaling complex together. So you can see this in this example here in figure 7.3, where we have a protein kinase in blue that adds these pink phosphate groups to scaffold protein. Then these phosphate groups allow for different other proteins to bind. So you can see here, this green scaffold then brings together these different proteins so they can function together. So you might get a membrane localization protein, so this helps bring the whole complex to a specific location. Then you can also bring together enzymes and substrates and promote conformational changes in these proteins. So scaffolds essentially form a backbone for these signaling components to come together. Another example of a multi-protein complex are adapter proteins. So adapter proteins are a little different from a scaffold. Here they essentially link different proteins together, usually two or more proteins. So here we can see the adapter here, the GRB2 protein, which binds to signaling protein through the SH3 domains, which are similar to the SH2 domains. And then this allows activation to occur. And the GRB2, our adapter protein, will then bind to the phosphotyrosine here that's been added to the receptor via one of these SH2 domains. And this brings our tyrosine kinase protein to the location of the receptor. So scaffolds and adapter proteins are similar, but they act in slightly different ways. The scaffold binds multiple different components so they all can act together. The adapter helps link specific things together. So another component that's often found in these signaling pathways are small g proteins. So these act essentially as molecular switches. And you might have heard receptor GTPase or G protein coupled receptors. And these GTPases are distinct from G protein coupled receptors. They're typically small molecules, proteins, that act as switches. So they bind either GDP or GTP. And GTP is the active form. And then we have associated proteins that help with the function of these GTPases. So GDP can be exchanged for GTP by what's known as a GEF, or a guanine nucleotide exchange factor. And some examples of this, one that you might have heard of previously, is called RAS, which is involved in cell proliferation. So these GEFs cause the release of GDP, and this allows for the binding of GTP. And that's because GTP is typically found in higher concentrations in the cell. So it typically will preferentially bind to the GTPase. And this leads to a conformational change and effector activity in this GTPase. And the GTPase activity will eventually convert that GTP into a GDP just through its constitutive enzyme activity. The GTPase activating protein, or GAP, helps convert this GTP to GDP conversion. And this helps regulate the activity of the GTPases. So here we have an example where in the resting state, a small G protein is bound to GDP and is inactive. And you can see that here. And then signaling activates this guanine nucleotide exchange factor, these GEFs, to help increase the exchange of GDP to GTP. And you can see here GTP is represented with these three pink phosphate groups, since it's guanine triphosphate. And GDP is represented with the two pink phosphate domains, since it's guanine diphosphate. So then When bound to GTP, this small G protein is active as an effector molecule. It can interact with and influence other proteins. However, over time, the enzymatic activity of the GTPase returns this to GDP, where it is not as active. So the activity of GTPases is determined through this conversion between GTP and GDP. So then, with these different components, our signaling proteins may be bound to the cell membrane in several different ways. So we talked about scaffold proteins, so that's one example, and this helps lead to the recruitment of other proteins with SH2 domains. The receptors that are in the membrane can also bind these small GTPases, which then bind additional signaling proteins. And then finally, we'll talk a bit more about this in detail, but modified lipids in the membrane can also affect which proteins are bound. So you can see this represented here in this figure. So binding to phosphorylated sites on the membrane-associated protein brings these different important signaling modules together. It's also possible that these small GTPases can act on different signaling proteins. And then, as I said, we'll talk a bit more, but this PI3 kinase will phosphorylate a lipid called PIP2 to generate PIP3. And you'll see a lot of the functions downstream of PIP3. It's a very important signaling module. It's also possible that these proteins can just bind to different membrane lipids. Another way that signals are modulated is through post-translational modifications. So I already mentioned one, which is phosphorylation through kinases, but other potential post-translational modifications include dephosphorylation as well as protein ubiquitination. So ubiquitination is a way that protein degradation is modulated. So ubiquitin ligase is an enzyme that transfers these ubiquitin proteins to covalently attach to a different protein. And this marks that protein for degradation, typically by the proteasome. And the location of the attachment can actually have different outcomes. So ubiquitination to lysine number 48, or K48, targets this protein for degradation at the proteasome, whereas ubiquitination of membrane proteins can target them for degradation through the lysosome pathway. So now that we've gone through a few components of different signaling pathways, we'll focus specifically on how antigen receptor signaling leads to lymphocyte activation. So these signals they consist of a variable chain which recognizes antigen and an invariant chain which helps with the signaling pathway. So you can see here the T cell receptor. So you guys talked about in previous lectures about this TCR. And you can see here that it really doesn't have much of an intracellular signaling domain. So it has to bind to other proteins that have more extensive intracellular domains in order to propagate a signal that leads to T cell activation. So it does this by binding different invariant chains. So that would be CD3. And there are several different CD3 chains, including gamma, epsilon, and delta, as well as binding what's called the zeta chain. And here you can see this in green. And the signaling of these proteins is mediated by domains known as ITAMs. And ITAM stands for immunoreceptor tyrosine-based activation motif. So B cell receptors act very similarly. So instead of CD3, they associate with Ig beta and Ig alpha, which again have ITAM signaling domains. So when antigen is recognized by the TCR, this produces a signal across the cell membrane. And this allows T cells to respond to pretty low levels of antigen binding, so less than 50 peptide MHC complex interactions of the whole cell. And this peptide MHC, like you guys talked about in previous lectures, binds the T cell receptor. The activation, the exact mechanism of activation is still a little unclear. So some possibilities are that in addition to the TCR binding foreign peptide MHC complexes, it also binds self-peptide MHC complexes as well. This would be at a low level, but it's possible that you need the combination of these different bindings to activate the T cell. Another possibility is that binding leads to conformation change in different proteins, or that the TCR receptors form microclusters. So they group together in small clusters, which then associate with clusters of other signaling components. And finally, we'll talk about this a little bit more later, but it's possible that actually TCR binding to a peptide MHC complex excludes inhibitory proteins that are too large to be located near the TCR. So antigen recognition by the TCR and its co-receptors, CD3, leads to phosphorylation of ITAMs. These co-receptors also include CD4 and CD8, which you guys talked about in relationship to helper T cells or cytotoxic T cells. And here, the intracellular domains associate with non-receptor kinases. So these are those kinases that are not part of the receptor protein, but they still come in here and bind to the intracellular domain of those proteins. The co-receptors, such as CD4, CD8, also come in and bind the MHC. And this helps stabilize this peptide MHC TCR complex. So often this association here between the MHC peptide and TCR is quite transient. It binds and disassociates pretty quickly. And so in order for signaling to occur, this association needs to last longer. So that would allow these tyrosine kinases more time to phosphorylate the proteins. And the co-receptors come in and bind to MHC class 2, and this helps slow down the dissociation. It essentially helps those peptide MHCs and TCR to stick together longer, which then leads to more phosphorylation and downstream signaling. And I mentioned earlier that it's possible that inhibitory molecules might be size excluded. So LIC, which is an example of the tyrosine kinase, is typically actually deactivated by another membrane protein called CD45. And this can help counteract the positive signaling here. So if CD45 is located near this TCR receptor complex, it will counteract and block signaling in the cell. So it's actually CD45 is a very large molecule, so it cannot fit in this area here between the two cell membranes. So the fact that the TCR and MHC class 2 binding occurs actually may force out that CD45 molecule, so then there's nothing stopping LIC to phosphorylate these ITAMs intracellularly. So once we have these ITAMs phosphorylated, they recruit and activate different adapter proteins. So one really important example is ZAP70. And ITAMs actually bind a phosphorylated and activated ZAP70. So ZAP70 here, shown in this yellow bent molecule actually has multiple SH2 domains, and these here, shown bound to two pink phosphorylases, can bind to the ITAMs. And then when bound, Lick, shown here, is able to additionally phosphorylate ZAP70, which allows for it to become active. And this is shown here. It's a little more clear. So typically, ZAP70 is in its inactive conformation, which you can see on the left. So here, when these SH2 domains are not phosphorylated, the ZAP70 protein cannot bind to an ITAM. However, when ZAP70 interacts with phosphate groups on an ITAM, this allows for it to become activated, and Lick can come and phosphorylate this kinase domain, activating ZAP70. So these phosphorylation and binding events lead to a change in the shape of ZAP70, which allows it to have its kinase activity and mediate further downstream signaling. So we see similar binding motifs in other cellular receptors. And so these ITAMs can also be found on receptors on NK cells, macrophages. A good example of this is the Fc gamma receptor 3, which you can see an example here. It also is found associated with different other receptors on NK cells, as well as mast cells and basophils. So these ITAMs are a motif that are found across many signaling domains.